Hereby I open this academic ceremony in which Miss Katherine Bassil will defend her academic thesis entitled Stress in a Dish, Modeling the Neurobiology of Glucocorticoids in Vitro, Investigating Stress Susceptibility and Highlighting Ethical Implications. May I invite the candidate to present a summary of your studies and the conclusions of your thesis in the next 15 minutes. Thank you very much.
Thank you very much. Dear Prorector, dear members of the Corona, dear family, friends, and colleagues, thank you for being here today. Sorry. In the upcoming 15 minutes, I will present you with a summary of my thesis and the research that I've conducted during my time as a PhD. This is my daughter Mo, a happy, healthy, and silly toddler. She was born in the Netherlands in a peaceful and enriched environment, showered with love and attention from all those around her. And this is a 5-year-old Syrian child. He grew up during the Syrian civil war and can be seen in this picture moments after he was rescued from a demolished building that was hit by an airstrike. Early life stress in the form of child abuse, accidents, and traumatic experiences during war is a major risk factor for stress-related disorders, including depression and post-traumatic stress disorder, or PTSD. Think of a building as the timeline of an individual's brain development. The foundation of that building represents the early stages of brain development. A weak or damaged foundation represents the susceptibility of this building to earthquakes, for example, while a strong foundation is necessary to ensure a sturdy and resilient structure. Similarly, early life stress can weaken an individual's ability to cope with stressors early in life, increasing their susceptibility to stress disorders later in life. Early brain development is a critical period that is increasingly vulnerable to stress, particularly in the prefrontal cortex region of the brain. During this period, neurons or brain cells, the building blocks of the brain, are being generated, forming connections, and establishing their functions, which will eventually impact the regulation of thoughts, emotions, and actions. But what exactly happens when you are exposed to stress? Upon exposure, the stress response is activated, bringing about a number of changes in the brain that eventually leads to the release of the stress hormone, cortisol. Similar to a domino effect, stress activates a series of changes, eventually leading to the fall of the last domino, which represents the changes caused by cortisol to the body and brain. Now, knowing that stress impacts the developing brain, and particularly through the release of cortisol, we were interested and we asked ourselves about the biological changes caused by glucocorticoids like cortisol in neurons. So in chapter 2 of my thesis, we provide an overview of different studies that have investigated this question. Now, the hormone cortisol can have different effects within neurons, and depending on its quantity and frequency, the outcome may be different. And as you've probably heard before, stress is not necessarily bad. Think of the stress levels that I'm going through now. In small quantities and for short periods of time, the release of cortisol might actually help neurons adapt to stress. Whereas at high concentrations and for longer periods of time, these changes become detrimental, leading to serious changes in the morphology and function of neurons, even leading to their death. However, the biological effects of high cortisol have not been extensively investigated in the prefrontal cortex, in human cortical neurons in particular. And this is very relevant because this region of the brain is not only susceptible to stress, but it has been also associated with abnormalities in patients that suffer from stress disorders. So we were interested in understanding the biological effects of excessive and chronic cortisol in human cortical neurons at different stages of neurodevelopment. But how do we do this? For this aim, we used a 2-dimensional culture model. Now, 2D models, including stem cell-derived neurons, as the name implies, are derived from stem cells. They can be generated by collecting embryonic, skin, or blood cells from humans, and these models consist of growing and differentiating neurons in a plastic dish, supplemented with nutrients to ensure their growth and maintenance. These are typically used to study biological processes, but also to test the effects of hormones and drugs. Now, in chapter 3 of my thesis, we took human neural stem cells and differentiated them into neural progenitor cells by exposing them to specific chemical compounds that will shape and transform these stem cells into more brain-like cells. Now, these progenitor cells were then further differentiated or specialized into young, immature neurons for a few days. These neurons were then left to grow and differentiate or specialize even further into maturing cortical neurons for a few weeks. At each of these 3 stages, we exposed the cells to high concentrations of cortisol chronically, and so we ended up comparing unstressed cells with stressed cortisol-exposed cells. We made use of different experiments to examine these effects, ranging from microscale changes, looking at the DNA itself, to more macroscale changes, looking at overall morphology and function. At the first stage, neural progenitor cells exposed to cortisol showed a decrease in cell proliferation. Now, this is a function that is used by cells to grow and expand. It can be seen in this illustration by a decrease in the number of red cells, which represents actively dividing cells. We also observed a decrease in cell survival, which can be seen as a total decrease in the number of cells. We saw an increase in early stages of cell death. This can be seen here as those dark cells that are slowly deteriorating and which will be followed by their death. At the second stage, young, immature neurons exposed to cortisol showed a decrease in a gene, PAC6, that is involved in the formation of new neurons. At the third stage, maturing cortical neurons exposed to cortisol showed an increase in PSD95, and this is a protein that is known to be involved in processes that strengthen synapses or connections between the brain. We saw a decrease in neuronal activity, which was represented here by a decrease in the number of peaks, and we saw a decrease in GFAP, which is an expression which is a gene that is typically expressed in cells that support neurons, and these cells are very important for the survival and functioning of neurons. Now, altogether, these results highlight the negative effects of chronic and excessive cortisol in human cortical neurons on their survival, their differentiation, and their maturation. We then wanted to ask the question differently. We wanted to know, of all the genes that are driving this differentiation process, which ones are most susceptible to cortisol? And so in chapter 4 of my thesis, we ended up identifying several cortisol-sensitive genes at different stages of neuronal differentiation. In the early stages, we identified genes that are known to be involved in the formation of synapses, with synapses being the contact point where 2 neurons meet. Whereas at later stages, we identified genes that are involved in the excitability of neurons or the flexibility of neurons to environmental changes. Now, with that being said, the findings of my research leads us to partially understand the difference in brain development between these 2 kids. The effects of early life stress on neuronal development caused by high cortisol levels may lead to less neurons, but also may weaken connections between neurons in the brain, which will more likely or could explain why these individuals that are exposed to stress early on develop disorders later in life. Now, the image that I've previously introduced to you only represents half of the truth, because as a matter of fact, not all individuals that are exposed to stress or trauma end up developing a disorder. We would all be suffering from a disorder if this was not the case. While some are susceptible, others are resilient and do not show any symptoms. And to make things even more complicated, we really don't know why. Given the importance of understanding susceptibility and resilience, the next question was, can we investigate this in neurons in culture? In chapter 5, we investigate the effects of our established cortisol model on a potential biomarker for PTSD susceptibility. Now, as this gene was previously shown to be dysregulated in soldiers that are susceptible to PTSD, the question was simple. Will this gene be responsive to a cortisol treatment in neurons? Now, the reason for the dysregulation of this gene is DNA methylation. This is a biological process that influences whether a gene will become active or inactive. The more methylation, the gene is blocked and more likely to be inactive. The less methylation, the more active. As expected, cortisol exposure did lead to a decrease in methylation of DOS22. As we could say, this was responsive to a stress challenge in culture in neurons. Now, while this is merely a pilot study, the finding does point towards the opportunity to study similar genes in neurons, in culture, and to better understand how they are involved in susceptibility to disorders. Now, two-dimensional cultures, like the one I used for my research, are not the only models out there to study neurodevelopment. In recent years, the generation of more complex models has really gained a lot of attention. Cerebral organoids in particular have really stolen the spotlight. But what are cerebral organoids? They are stem cell derived, three-dimensional brain tissue, only a few millimeters in size, and are more similar to the human brain than two-dimensional cultures. In fact, they have even been shown to be comparable in some aspects to the brain of a second trimester fetus. In general, they are simply better at mimicking certain aspects of human neurodevelopment. The uses of cerebral organoids ranges from personalized drug testing, studying brain development, or even better understanding the underlying mechanisms in brain diseases. However, what if I told you that these complex models are associated with ethical challenges? So, despite great advances in cerebral organoid technology, this research has been accompanied with a lot of ethical debate. While many experts have raised concerns about potential consciousness in these organoids, others have argued that there are more important ethical issues that we need to be addressing. For this reason, in my thesis in Chapter 7, I highlight and focus on those more pressing issues, particularly in relation to stress research. First, there are several types of cerebral organoids, but also several ways of generating them. And so I argue that ethical discussions should separate between those that are ethically problematic and those that are not. This is important to avoid any unnecessary regulation that would impact all organoid stress research. The reason we do not want that is because organoids really offer a lot of promises for patients. Second, communication about cerebral organoid research and the ethics should be carefully conducted when recruiting potential donors and research participants. The media has often made claims about cerebral organoids, saying things like they are sentient, that they are intelligent, even comparing them to brains in a dish. Now, these claims are not only misleading, but can discourage the public from participating or even supporting such research. So therefore, it is important to be very careful when sharing results of similar research to avoid spreading misinformation, false hope, and hype. And finally, research with animal chimera. Now, these are animals that are transplanted with human brain organoids, which means that their brain is composed of both human and animal tissue. Now, their use in stress research should really be considered carefully because this is not only a very invasive procedure for these animals, but stressing these animals might induce more pronounced stress-like symptoms, which could seriously negatively impact the well-being of the animals. Now, to conclude, during the first part of this thesis, I show that chronic and excessive cortisol leads to negative regulation of human cortical development. I also discuss how culture models can be used to investigate susceptibility genes. However, there remains a lot of work to be done, not only to improve the reproducibility of similar studies, but also the relevance to patients in the clinic. In the second part of this thesis, I highlighted some of the ethical considerations that accompany the use of advanced 3D brain cultures, and specifically in relation to stress research. And overall, I hope I therefore convinced you that simple culture models allow the study of stress mechanisms, and that this type of research also comes with ethical implications that require the consideration of scientists, ethicists, and the media alike. With that, I would like to thank you all for your attention, and I give the word back to the pro-rector.
Thank you very much, Ms. Bassil. Thank you very much, Ms. Candidate, for your very clear presentation. And I must compliment you also with your illustrations. They were very nicely illustrated. Compliments. We have now, for the members in the audience, we have now 6 opponents who are going to address questions to the candidate, and one of the opponents will be online. But we're going to start with the on-site opponent, which will be Professor van den Hove. He is professor in neuroepigenetics at our university, and I'd like to give him the first word for some questioning.
Thank you very much, Mr. Prorector. First of all, my compliments to you for this very nice thesis. Content-wise, also the outer appearance. Although I have to say, this neuron on the right, it's a bit devilish, the appearance. I always get distracted by it, but okay. No, my compliments also to your promotion team on this very nice thesis. And also my compliments, I think, for the academic initiative you have taken outside the scope of the direct scope of your thesis. Let me first ask you maybe some questions before we drown in compliments. My first question actually is on the genes that you find enriched or altered in terms of their expression after glucocorticoid exposure. So you actually state, or you observe that actually these genes seem to overlap more with gene and candidate genes for schizophrenia or psychotic disorders than for stress-related disorders like PTSD and depression and such. Whereas the scope of your work was mainly addressing those genes. So what does that make you conclude is, let's say, my first open question. Is that related to the overlap in phenotypes between these different disorders? Do you think you, in hindsight, then should have been using another model? What's your view on these findings?
Dear highly esteemed opponent, thank you very much for your kind words and for this very important question. I think I will kind of break it down in different aspects. Firstly, starting with the model itself. I think we need to, every time we make use of a certain model to investigate a certain research question, we should make sure that we look at both the advantages and limitations of the model. The model that I made use of, which are stem cell-derived neurons, these are better, their use is mainly made to replicate certain neurodevelopmental aspects. And I think the reason why we were able to kind of see more of similar mapping with schizophrenia, for example, or other neurodevelopmental disorders is mainly due to the model itself. It is really representing an early brain developmental stage and not looking at, for example, an adult stage where we most often diagnose people with PTSD and depression, for example, and where most of the research that's being done is being done on those at the adult stage. So I would say the model itself does kind of create this, let's say, limitation in the sense that we are not modeling stress-related disorders per se and only, but we are modeling more of a category of disorders where stress is a risk factor or a common denominator for them all. Yeah, I think this is the way I see it initially, and also the aim that we had with kind of making use of that model, but also exposing it to cortisol, is simply to create a stress model or a stress challenge in vitro in order to kind of create a certain stress challenge and see how the neurons are going to respond to it, and not specifically kind of model PTSD or traumatic stress per se, but really more to have a general cellular stress that is mostly resembling what is happening in the brain.
No, very clear, thanks. For a second question, may I ask one of your parents to read out statement number 1, please?
In vitro glucocorticoid models are used to investigate stress-related neurobiological mechanisms and disorders, but standardization is needed for improved study validation and replication.
Thank you very much. Yeah, the same claims have been made also for animal models, standardization and such, although there's also people that sort of oppose this concept, saying that for example, Kaplan, when it comes to animal models, said that the danger is not working in with too many models, but actually with too few. So my question is standardization in this case, do you really see that as a solution to the problem? Because if we look at let's say, real-life problems when it comes to glucocorticoids. It's so complicated. I mean, we have different cell types, different types of neurons, the interneurons. We actually have different brain regions interacting with each other. Then you have chronic, acute, all the things you actually also described in your thesis. Do you really think that standardization is a solution there? Or do you see other ways around that?
A very important question. I think if I will direct the attention maybe a bit to chapter 2, because this is where this conclusion kind of emanated from. In chapter 2, we really conducted a review where we looked at a variety of different in vitro studies that have investigated the effects of glucocorticoids in neuronal cell types, or let's say central nervous system cells. And what we see here is a big variety in more of the, let's say, experimental conditions and not per se the model that they are trying to set up. So we are not talking about having an acute model and a chronic model. We see already within the acute stress challenge that they are trying to replicate or to model in vitro, we see a lot of variability there. We see variability in the concentrations that are being used. We're looking at the lowest concentrations being used in the nanomolar to the highest concentrations being in the millimolar. And I think this is quite a very big difference when we're looking at cell responses and how the cells respond to different concentrations. And I think when I talk about standardization, I think it's more towards really focusing on certain parameters, certain acceptable parameters that the field should really adhere to when doing such research. And when I talk about standardization, I also talk about defining acute and chronic stress in vitro. I think nowadays they are kind of used also with a variety of variables. And for example, acute stress is it 1 hour? Is it 6 hours? Is it 24 hours? And I think when we look at in vitro studies, there is a lot of contradictory results when it comes to these studies, especially if we're looking only at the acute stress models that are being created. And I think so. More, if I want to answer the question in like a very summary, in a quick summary, is I think maybe the experimental conditions need to be more standardized and not so much creating different types of models.
I think I agree, because indeed it's actually already difficult to replicate the same experiment in the same lab with cultured cells. I'm very happy with the answer, and I give the word back to the pro-rector.
Thank you very much. The opposition will be continued by Professor Illes. She is affiliated with the Department of Neuroscience and Neuroethics at the University of British Columbia, and of course I would like to welcome her that she came all the way from Canada to visit us and to attend this meeting. I'd like to give you the word.
Thank you, Rector, and good afternoon, Ms. Bassil, and good afternoon to all of your supporters in the audience. And I sit on this side of the room perhaps as an accidental symbol of my support for you, and what a pleasure it has been to see you evolve over the past 10 years. And I join our colleague in congratulating you on some work very well done. I do have some challenging questions that I'd like to pose. And the first has to do with biomarkers. In a world today in which we are faced with AI and neural networks, hijacking of data, unexpected interventions, adverse consequences, unintended consequences, all related to our computer world, our fast-paced computer world. If we start biomarking for susceptibility to diseases, conditions like PTSD, how do we avoid putting people at greater risk than actually benefiting them?
Dear highly esteemed opponent, thank you very much for your kind words and for this very timely question. I think as we are looking at the past decade, we are seeing a lot of technologies that are really rapidly evolving. And it is sometimes, I think, a shame that the ethics is not developing as rapidly enough as the technology itself. And I think when thinking of biomarkers for susceptibility or even resilience, we need to be having this question now. Even though the technology is not already being used in the clinic with patients or in certain contexts, specifically if I think of, for example, the military context, where when we're talking about PTSD susceptibility, this is where the intended use of this technology is needed. we should be having these conversations because identifying individuals that are susceptible or resilient could bring about a number of ethical implications that we probably do not see at forehand because we think that the technology is there to do good, and that is to prevent cases of PTSD, to decrease the economic burden that is associated with PTSD. But I think in chapter 8 of my thesis, I do reflect a bit on that question. And I think it is really a question that should be regarded not only in the potential uses and benefits that biomarker technology offers or brings to the table, but really what are the unintended consequences that may arise from such a usage. And if I could give a few examples, identifying individuals that are susceptible to PTSD on one hand may decrease the stigma, of course, but on the other hand could also increase the stigma because certain individuals will become labeled with being susceptible to PTSD. And then this brings me to what does that mean for their colleagues that are, for example, labeled as resilient to PTSD? How will the discrimination within the workplace, particularly in military context where we already know there are a lot of forces, there is already a lot of stigma and discrimination taking place within the context of the military, how will using biomarker for identifying susceptibility or risk profiles really exacerbate these issues?
Very good, well done. A second challenge for you, and perhaps this is a false challenge, but nevertheless, you've shared with us in your presentation today and in your thesis a call for action for neuroscientists to become more engaged in science communication, engagement with patients, with the public. How do we prevent the neuroscience community from itself becoming incredibly stressed out? From now layering on these additional very heavy responsibilities for which neuroscientists today are not actually really well trained?
Thank you for this question. I think I will maybe go back to the discussion that we had earlier this morning in the symposium about integrating neuroethics in education. And I think it starts there. If there is a certain awareness that is brought to the attention of neuroscientists about how they should communicate their findings, how oversimplification is not always the right way, I think starting there could be a very good place. It could be a very good place to start because I think simply sometimes bringing that to the attention of individuals could really make them more aware of what are the things that they should be more attentive of when, for example, having press releases about their research. And of course, the neuroscientists may argue and say that we already have a lot to do, and this is probably not our task, but the task of science communicators or people that are responsible to kind of break down the science and make it more comprehensible. But I think it's really a teamwork. It should be I think all parties, and I mentioned the media as well, and science communicators are also responsible for conducting responsible communication with the public, because this is the results that are emanating from research and science also impact the public and also impact other stakeholders. One thing that I think is very important, and I will just briefly mention an example to showcase how serious this is. If we think of embryonic research and how this has been impacted by the public discourse and the media kind of also shifting directions in certain, or shifting the conversation in certain directions that could actually be very harmful to society. So I think we should learn from our mistakes and do better and start by learning and introducing that in the educational aspects. Thank you.
Well, I have one more for you at this time, and it's a shift away from the ethics, more into the neuroscience. In statement number 3, you declare, I suppose, schizophrenia as a neurodevelopmental disorder. And I don't conventionally think of schizophrenia as a neurodevelopmental disorder. And I was wondering if you could defend that statement for us, please.
I think, of course, there are different ways to look at that, and why I would let's say put schizophrenia in let's say the category of neurodevelopmental disorders is because many of the dysregulations that we are seeing are at the level of brain development, and we see already early on in several individuals that are suffering from schizophrenia, we already see perturbations and dysregulations in, for example, the maturation and synaptic formation, in the way the brain is forming connections. And I think in that aspect, we could put it in the category of neurodevelopmental disorders, but it should not be exclusive to that category. And we should think about it, of course, as probably also, I would also even consider it as a stress-related disorder, because stress is a very important risk factor and really kind of increases the risk of individuals that are exposed to stress and have, let's say, an underlying genetic vulnerability to develop schizophrenia later on.
Thank you very much. And I also would like to thank you because you were the only member today available here as the member of an assessment committee. So we move forward, and we have moved forward to Professor Vermette. Professor Vermette is professor in medical biology and psychiatric aspects of psychotrauma at the Leiden University Medical Center, and I also would like to thank him that he came today all the way from Leiden to attend this meeting over here. I'd like to give you the word.
Thank you, dear Rector. Dear candidate, congratulations on a wonderful dissertation and a beautiful book. I had some psychedelic associations with the front cover, but maybe more me than actually what you tried to put into it. No, it's beautiful to read and wonderful how you illustrate the dissertation with graphical illustration. As I was reading it, I had some reflections of the early work of Ron Duman that you may have come across, who showed hippocampal neurons in a dish and then did some SSRIs and some things, and then he showed neurogenesis. That for years that has led research into a new metaphor, like neurogenesis can rewire the brain, so we should think of new life and learning and growth modeling. I also like, and then I'll start my questions about chapter 7 and chapter 8, if we have time, on what you just nicely said about integrating these two fundamental and more neuroethics approaches into one dissertation. Okay, let me just be bluntly and sort of integrate first the chapter on number 7. How can a scientist, like a neuroscientist like you, with a title like Stress in a Dish, make such broad implications about psychiatric disorders and susceptibility and like these things that have far-stretching clinical implications?
Sorry, that have your...
Far-stretching clinical implications.
Dear highly esteemed opponent, thank you very much for your kind words and for this question. I think in vitro models have a lot to offer. And if it's not in the immediate sense that they can immediately be translated to the clinic, I think they offer a lot of insight into the mechanisms that is conferring susceptibility to individuals that are suffering from stress disorders. And I think what we've learned from all the in vitro work that has been done is that we have been able to identify certain biomarkers or certain key players in the regulation of the stress response. And we have been able to also differentiate between individuals that do end up developing stress disorders and those that do not. And that is also partially thanks to in vitro research and work like my own. I think, of course, we always need to be cautious about making bold statements about how this will impact the clinic and patients themselves. Sorry.
No, please, because what I heard you say, mechanisms, right? I feel that also, and it's like what we can engage in this a little bit of discussion as well. You, as a scientist, have moral implications about what you're saying to whom, with what kind of imperative, right? So my question actually was about in chapter 7 there. You are addressing a committee, a guiding committee, and you just said a scientist and ethicists and the media need to be part in that. Who else needs to be part in that guiding committee?
I think the public as well. I think the public has also a very important role in sharing their opinion, sharing their feelings about this kind of research that is taking place. And I think we've often forgotten the public, and we've often forgotten how they can also bring a very important contribution to the discussion about how far could research go? How far should...
Okay, let me interrupt you there.
Mm-hmm.
How far can it go?
I think in research, typically we're also always looking at weighing benefits versus risks.
Sure.
And I think this applies also to cerebral organoids. If the benefits outweigh the risks, it is of my opinion that we should proceed with such research.
I would have loved, and Lo, I am adding to what you just said, I would have loved, or maybe that could be a future exercise, that you have a couple of these vignettes, like what if this? And let's engage students and the public in a discourse, and then what if this? And then what if that? I think it's a great course for students to engage in that kind of provocative, like what is the limit for us? Where are we, a red line?
Definitely.
Second question is, you showed this young boy, right, who we all know that you just illustrated. I've been in the military, and early identification is utmost at our scope. So the vulnerable phenotype on the one end, and early identification on the other side. Like you nicely said, like, what is the pro and the con of identifying this boy as maybe prone for developing PTSD? Yeah, my question is more throwing it back to you. Should we, or do we have actually a profile for this young boy? Or do we have actually biomarkers at this day and age to identify who's susceptible for PTSD based on the material that we have now identified on your research?
I think we do have, of course, there's a lot of literature on certain phenotypes of early life adversity. And I think we typically, of course, look at, for example, typically what is being used in the field of PTSD and MDD is circulating cortisol levels, looking at the reactivity of the stress response to, for example, a stressor or a stressful challenge. But also if we look at, in general, or already being exposed to early life adversity is already kind of filling your profile as being more susceptible to stress disorders later in life. And of course, the typical symptoms that are associated with PTSD and in general stress-related disorders.
And if we go the other way, like you also said in chapter number 8, you talk about resiliency, and I don't know whether you named the word hardiness or so, resistancy for PTSD. Is that also something that you could drive from your DISH research to kind of a statement about resistancy? Or would that be a red line now? We can't go that far.
No. I think we could definitely do that. And I think in vitro also offers these promises because we can make use of, take samples from individuals that are susceptible, that we know are susceptible, and those that we know are resilient, and kind of create or generate neurons in addition, and also look at how they respond differentially to cortisol. And I think this would be one way of identifying certain resilience mechanisms to a stress challenge in vitro, and then see if how can this be brought to the clinic and translated to the clinic.
Now, very briefly then I'd like to challenge you on page 252. I mean, the paragraph starts with the end of guilt. Now, you're talking to a psychiatrist here, and I deal with shame and guilt in a lot of cases when it's about PTSD. Are you sure we can go there?
I think it's a question worth asking. I think it's a question worth asking and engaging with, and also seeing how patients feel about that question, or individuals that are in the military. If we are to tell them we have the tools to identify you as being resilient, would you want to know? How would you deal with such information? And I think this brings us back to the discussions we've been having about genetic testing, and this has also raised a lot of those questions. If you are susceptible to cancer, if you are susceptible to Alzheimer's, do you want to know? I think these are a very important question to ask and raise.
Thank you very much. I like the stamina, and I like your podcasts, and I think these are very important questions to be addressing to students, to the media, to ethicists, and to scientists like you. Thank you very much.
Thank you.
Give the word back to the rector.
Thank you very much. The opposition now will be continued by Dr. Daskalakis. He is assistant professor of psychiatry at Harvard University, and of course I would like to thank him also that he joins this afternoon. To you the word.
Thank you. Correction, I'm associate professor since 2 weeks.
I'm just reading my text.
Geachte kandidaat, I want to congratulate you for a very nice thesis, scientifically, also aesthetically, and also bringing the other aspects beyond the explicit experiments. I have prepared some challenging questions. First one, the transcriptomic effects you describe in chapters 3 and 4, that you derive this based on analysis using high levels of cortisol in vitro. Are they mediated by glucocorticoid receptor or mineralocorticoid receptor or both? And what additional experiments would you design, theoretically speaking, to test that?
Dear highly esteemed opponent, thank you for your kind words and your questions. I think for the first part of the question, yes, we have made use of a high concentration of cortisol. We know from literature, we know from the biology of how stress or cortisol impacts cells. We know that at low concentration levels, the MRs, or at basal levels, the MR, the mineralocorticoid receptor, is the main receptor that is active. Whereas at higher concentrations, the GR also becomes active. And this is very important. This interplay between the GR and the MR is very important to also lead to the stress response to kind of become inhibited and go back to baseline levels after that the stress has been has disappeared. And so looking at previous work, and specifically the work of Anake Real, where they really look at those differences in concentrations, low concentrations and high concentrations being 100 micromolar, similar to what I have used in those chapters, we see that through their work, it is the high concentration that is really the effects on the neurons and on neuronal differentiation and astrogliogenesis is taking place via the glucocorticoid receptor. However, of course, this is an assumption based on previous work on a different cell type. So going to your next question, I think the follow-up experiments to this would be to look at playing with the GR and MR using agonist and antagonist, and also seeing if we are able to see similar results, and based on what the activation of what receptor would we see similar results. And I think only then can we really come to a conclusion that these effects that we are seeing are GR mediated or MR mediated.
Thank you. Very satisfied by your answer. I go to my next question. There was the statement 3 in your statements that attract a lot of attention today. I want to ask a question about it from a different angle. So in this particular statement, you make a conclusion about the colocalization of cortisol-sensitive genes with genetic risk for schizophrenia, and and you are using an approach that is driven by using EQTLs and published GWAS of various disorders. But in chapter 5, you drive another conclusion by using EWAS results from PTSD only, top EWAS results from PTSD only. So my question comes to, from the genetic standpoint, PTSD genetics are, our genetic makeup that helps us respond to the environment. I wonder is, in the first angle, using EQTLs, the EQTLs used, or are they rather static? What kind of EQTL database you would envision to really test the overlap with PTSD genetics?
Thank you for this question. I think, indeed, we have taken a different approach between chapter 4 and chapter 5. And I think if we look at the colocalization that we did in chapter 5, it really focused, so we did look at PTSD, at MDD, and schizophrenia. And by not seeing a certain co-localization with PTSD risk variants, let's say, I think one thing that we could redo, for example, is instead of looking at published, for example, blood results or blood findings, right? Because these EQTLs have been identified in GWASs that were taken from plasma, from blood. I think it would be very interesting to look at postmortem data and see if there we could see some kind of overlap, because we are looking at neural tissue, and it would be also best to make sure we are correlating that with neural tissue, with brain tissue. And as such, I think I would maybe look more at postmortem and do that comparison there.
Mm-hmm. So you're basically saying that you would consider also not only focusing on EQTLs, but also real transcriptomic data from patients.
Definitely. And I think, of course, one of the limitations, I would say also of chapter 5, is that we are only looking at methylation and only looking at expression. And I think also looking at this interaction between risk variants and methylation or expression would be very interesting. And I think this would be in the context of taking sample like PTSD susceptible and resilient samples and see there if we can identify certain gene environment interactions. And I think this work has already been done partially by the group of Yehuda and Brennan, where they also looked at differential responsiveness to a dexamethasone challenge or cortisol in vitro.
Thank you. This is a very comprehensive answer. Thank you. If there is a time, I have another question. If not, I can skip it.
Unfortunately, there's no time.
Okay.
Okay, we have to proceed. We have to proceed, and we move now online. So we move to Professor Lesch. He's professor in molecular psychiatry, both affiliated with the University of Würzburg and Maastricht University, and I'd like to give him the word. Professor Lesch, I give you the word.
Thank you, Pro-Rector. Also would like to thank the candidate for this impressive thesis and the clear and stimulating presentation. My question concerns the potential and limitations of iPSC-derived cortical neuron cultures. And my question is a kind of extension of the question already posted by Professor van den Hove. You, in your thesis, you mentioned that you have a certain ratio of glutamatergic and GABAergic neurons. And these glutamatergic and GABAergic neurons are in an intricate balance, the excitatory-inhibitory balance. And this balance is very, very sensitive to intervention. It can already change the network activity when you insert the tip of your pipette into the culture dish. How did you control for this ratio and the network activity in your cortical neuron cultures?
Dear highly esteemed opponent, thank you for your kind words and these questions. Starting with the first question about the advantages and limitations of iPSC cultures. I think starting maybe with the limitations, iPSCs are not, the holy grail or the best thing out there. I think it's the best thing that we have so far to kind of better understand patient-specific phenotypes. And of course, it is very good at replicating neurodevelopmental aspects. However, the limitations is we still have a lot of variability between cell lines in iPSCs. We have also several variabilities that exist between protocols in generation of iPSC-derived neurons. I think these are some of the limitations that I think are on the top list of limitations when it comes to stem cell derived cultures. Whereas when it comes to the advantages, I already mentioned a few. They are really good at replicating certain aspects of neurodevelopment that we see in vivo. I think, of course, the more we look at the 3D cultures, we start seeing that has even better advantages. especially in the way the cells are organized, the self-organization that is typically characterized by cerebral organoids. And I think really this patient-specific phenotype that we can recreate and culture is very interesting, because we did not have anything like that before. And now we can, by taking samples from individuals that suffer from a certain disease, we can not only compare that with controls, but if we are talking about a specific, for example, genetic risk variant that is a characteristic of the disease, we can also create isogenic controls, for example, to compare how would altering this mutation, for example, improve the phenotype at hand.
Let me come back to this excitatory-inhibitory balance, which is so important across psychiatric disorders. Many of those have been suggested to have an imbalance of excitatory-inhibitory network activity. How can we, in the future, control for this variation in balance? What would you do if you would design new experiments to measure electrophysiological activity, particularly with the focus on EI balance?
Going on to the second question, I think, of course, if I look back and if we would want to disentangle these differential responses between glutamatergic and GABAergic, I think one thing to do is create pure cultures of glutamatergic neurons and pure cultures of GABAergic neurons, and then see how they respond to cortisol in vitro. And I think only then can we really see how this synergistic effect, once putting them together, is being impacted based on these individual experiments that we need to perform before that.
In this respect, I disagree with you. I'm sorry. The brain is based on network activity, and you need a kind of balance between excitation and inhibition. If you would disentangle this into single neurons, you would not get the full picture, not to talk about monoaminergic modulation of this EI balance. I would not suggest this, but I perhaps would suggest to do either patch clamping of neuronal activity, or even better, doing multi-electrode arrays and measuring this EI balance in a very, very dense or high resolution. Good. My second question also alludes a little bit on the work we did a few years ago in collaboration with Professor van den Hoven, where we showed that prenatal stress has a very strong impact on myelination or the expression of myelination markers. How would you address this in a future experiment to include perhaps glia cells like oligodendrocytes or astrocytes, given the fact that glucocorticoids modulate myelination markers?
Thank you for this question. I think going back to the advantages of iPSCs is that we can derive different cell types, and I think already in our group here, in the neuroepigenetics group at Maastricht University, they have been successful in deriving oligodendrocyte precursor cells. So I think this could be one way of doing that and investigating myelin formation and how because we know also, for example, that the myelin binding protein does decrease in response to cortisol and in response to glucocorticoids. So I think this would definitely be possible by making use of iPS cultures, and particularly differentiating them into glial cells like oligodendrocytes.
Thank you. I think all my questions are adequately addressed, and I would like to give back my word to the pro-rector.
Thank you very much. Then the opposition will be continued by Dr. Van Tienen. She is affiliated with the Department of Toxicology and Genomics at our university, and I'd like to give her the possibility also to ask some questions to you.
Thank you, Pro-Rector. Dear candidate, congratulations with a very nice thesis. Also, I would like to congratulate your promotion team. I would like to discuss chapter 3 and 4 with you to start. So in chapter 3 and 4, you successfully managed to differentiate embryonic stem cells into neurons and analyze them at different stages. one of these analyses was transcriptomics, and there you showed that cortisol pathway is activated at different stages. But you also did qPCR on the glucocorticoid receptor, which was unchanged. So can you elaborate, was that unexpected, or what do you think the cause is?
Dear esteemed opponent, thank you for your kind words and your question. I think this is definitely one of the main, let's say, unexpected findings that we had. But going back to the model itself, it really represents an embryonic developmental stage model. So we are really looking at glucocorticoid levels or glucocorticoid receptor levels during embryonic stages. And the literature does show us that glucocorticoid receptors are really very low expressed in these early stages, and it slowly increases as we move towards later developmental stages. And previous actually papers that were trying to do very similar work, looking at differential responsiveness to glucocorticoids, also had a similar problem where they were not able to see any responsiveness to glucocorticoid receptors. And I think one recurrent theme that keeps on coming is that this could be not the best model to kind of look for these changes because of these very low expression levels. However, by looking at, for example, downstream cortisol or glucocorticoid responsive genes, we see that there is an activation in those genes. So this gives us some, hope that our model is not completely, that we should not discard this model, but that we can look further. And I think the more we advance in this technology and really improve the ways and the protocols and ways that we are growing these neurons, I think we can better answer this question and see how we can improve it to better mimic certain aspects of the glucocorticoid signaling.
Okay. Yeah, you mentioned the concentration is very low, and also looking at the standard deviations, you see some variation between samples. Do you think the differentiation procedure is challenging and time-consuming, so lots of variation can be introduced between the cultures? Is that a source, of course? And which variation do you think is the most important?
I think definitely we are also looking at a heterogeneous culture that is not exclusively neuronal. We also know that the culture does, so the neural progenitor cells are also differentiated into some glial cells that we see in culture. So definitely there is variation that is created with such a protocol, and I think trying to amend that is by creating pure neuronal cultures to kind of disentangle potential confounders. And I think another thing to also keep in mind is we only looked at gene expression, and I think no changes at gene expression does not necessarily mean that there is no change in the functional properties of that protein. So I think also looking at protein expression and how this, for example, interacts with other regulatory proteins would be very important to better answer the question: is it really that the receptors are not very responsive to glucocorticoids, or whether it's really more of a translational mechanism? So what is happening between mRNA expression and protein synthesis that could be explaining this lack of observed changes in expression.
Okay, thank you. And then I also had a question about, also chapter 3 and 4, but also related to chapter 2, in which you described the effect of stress on the mitochondria and the neurons. So you elaborate on morphology changes, functional changes, yet in chapter 3 and 4, I did not see any mitochondrial changes in your data. So aren't mitochondrial changes that relevant in this model?
I think this was not necessarily the focus of my thesis. So I think if we had looked deeper into some of the key genes and key proteins and regulators of mitochondrial functioning, I think I would be better able to answer your question. But I think we know from the literature, which was represented in chapter 2, is that, of course, the mitochondria is impacted. There are glucocorticoid receptors in the mitochondria that are responding to a cortisol exposure. And I think looking deeper into these mechanisms would definitely kind of give us better understanding of how this is happening in human cortical neurons.
Okay, so in your transcriptomics approach, it's not sensitive enough then to the...
I think that would probably also relate to your first question, right? Are the glucocorticoids level, the receptor levels, are they enough to capture such an effect? And if already at the cell stage we are not seeing any major changes on the glucocorticoid receptors, if at the mitochondrial level we have even less receptors, this could even be even more challenging to capture certain effects. So I think I would definitely, maybe with genetic manipulation or expression manipulation of these genes, this could be one way of kind of side-tracking these limitations of such a model and see what are the effects of glucocorticoids on downstream signaling cascades.
Okay, thank you for your answer.
You are allowed to finish a short question, which you are allowed, you don't have to answer it shortly. So I give first the opportunity, would you like to make a short question?
You have one short question. You use in chapter 3 and 4 two gene expression analysis approaches, one pathway analysis and one cluster analysis. Why did you use those two different approaches for these different chapters, and which one do you prefer?
I think the reason we chose two different approaches is to showcase that we can use two different approaches to answer a similar research question. And I think this only provides more added benefit in looking at the question from different perspectives. And I don't necessarily prefer one to the other, but if I would say what I really like about the second approach, which is, for example, what we kind of showed in chapter 5 also, is kind of maybe identifying certain neurodevelopmental set of genes that we can also further use in future experiments when looking at susceptibility and resilience. Can these neurodevelopmental set of genes kind of become a hallmark for these early life adversity phenotypes, and can we kind of make use of that to better translate or find certain treatment or prevention strategies by identifying these neurodevelopmental set of genes?
Okay, that's clear. Thank you.
Dear Ms. Bassil, dear Ms. Candidate, the time appointed for defending your thesis has now definitely passed. Yes, you finished. The degree committee will now withdraw to discuss the quality of the thesis, but in particular the way you have defended your thesis this afternoon. I request that you and the company here available await the results of our deliberations and the return in this room. Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Traveling so far from future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Miss Bassil, dear candidate, still, the degree committee here present has assessed the quality of your thesis, and in particular the way you have defended your thesis, I would say, with full enthusiasm. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Rutten is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And therefore, I invite your supervisor now to take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Katherine Christophe Bassil, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
I would also like to mention there will be a second certificate be presented for the school Mental Health and Neuroscience, and therefore I give the word to Dr. Cadis.
Thank you. So, on behalf of the Graduate School of Mental Health and Neuroscience, I am entitled to hand out this official certificate. The owner of this certificate has shown that to have received adequate training in diverse academic skills and has gained relevant experience to become a successful and independent scholar. So, congratulations. So, dear Dr. Bassil, dear Katherine, on behalf of Laurence and Bart, myself, sincere congratulations with your PhD. It is the culmination of hard work during a long journey. And it all started about 7 years ago when you came to Maastricht to follow your passion for neuroscience. You were enrolled in the Research Master Cognitive and Clinical Neuroscience and where you studied fundamental neuroscience. Already during the first year, you quickly expressed your interest in the biology of psychiatric disorders, and particularly in the way that stress and traumatic experiences change the brain through epigenetic processes. And I did find a lot of emails back that you clearly expressed that interest. And you already clearly indicated then that you wanted to investigate that using stem cell-based models. As this was the main research team also of Bart, we together discussed possibilities for an internship, but at that moment we did not have experience in stem cell cultures. And since you were quite determined on that topic, Bart brought you in contact with the lab of Professor Gage, from which we had contact from the former professor and pro-rector here, Professor Steinbusch. So Professor Gage in the Salk Institute of Biological Studies in San Diego, which is a pioneering lab in this particular type of stem cell technology. However, this famous lab does not really accept students. And Bart and me were a bit hesitant at first in the whole idea. But when Katherine has set her mind to something, nothing, and certainly not hesitant supervisors, will stop her from making it a reality. So Katherine took a plane to San Diego to meet with Professor Gage herself. And of course, with your natural grace in presentations and advocating, as we have all witnessed here today, and which was also referred to in the debate of the corona just a minute ago, and you know how to build arguments and discussions, you were able to persuade Professor Gage in San Diego to offer you an internship position in his lab. You had to come back here first, of course, and you wrote a research proposal and concealed the deal that you would also learn all about stem cells in the Gage lab and come back here in Maastricht to implement the techniques. And by that, you actually had secured your PhD position here before there was even a project. As I said before, nothing will stop Katherine. So then you left for San Diego, but in the meantime, here in Maastricht, you had met your partner Joris. And the one things that the challenges accompanying long-distance relationships would stop Katherine, of course not. So Joris joined Katherine to San Diego. You spent about 9 months there in San Diego with Gage, and under supervision of Dr. Krishna Vadadarya, you learned all about the stem cell models. You did many experiments that led to your thesis, your master thesis, and after that you came back here and you actually established these stem cell-based cultures in our lab, in the Translational Neuroscience lab. This was, of course, pioneering work by its own, because no one here had this particular expertise, especially not culturing neurons. But again, nothing will stop you, and thanks to you, we now have this valuable experience, and it's taught to many other young researchers also present here that use this technology. I would also like to acknowledge that you had also advice, of course, from other researchers within the university. We'd also like to thank Dr. Florence Fantine and her team in this respect. As the title of your thesis states, your project here was Stress in a Dish, and while mimicking stress by exposing culture neurons to stress hormones seems pretty straightforward, nothing is less true. It is tedious work. It requires weeks and months to grow the cells with many complicated procedures in between. And even with doing everything by the book, it can still go wrong, and sometimes it did. But uncooperative cell cultures will not stop you, of course not. You learned from it, started over, and continued with experiments, even during many weekends and holidays. And even the corona pandemic, although it created several setbacks, could not stop you. In the end, you succeeded in gathering all the data and compiling this important thesis. It reflects the outcome of your lab experiments, giving insights in the effects of stress on the maturation of brain cells at the molecular level. But in addition, and as we also have discussed here, the reader will have seen that it also reflects your passion on the ethics of science. And in collaboration with Dr. Dorothy Horskutter, thank you to Dr. Horskutter, you examined the ethical and societal implications of using stem cells for brain research. But your ethics mind reaches far beyond this aspect. You have been a proponent in opening discussion about gender equality in academia and about the demanding working environment for PhD students. And also here, you show a relentless determination. Nothing will stop you, even not when famous established professors try to disparage you on social media. With these fights and all your other efforts in raising public awareness on the impact of neuroscience research and its clinical consequences, you have gained a deep respect from Maastricht's academic community. And even looking to all the things that you still have done on the side that are mentioned in your thesis, there were even many things that I wasn't even aware of. So that leaves me with the question, how were you able to combine all this? You were involved in so many things that sometimes you were a bit afraid that you would lose focus. And if that wasn't enough, in the midst of your project, Mo was born. And maybe becoming a mother and taking care of a little baby would stop Katherine. On the contrary, of course, it seems that it even brought you more energy and has strengthened your motivation to fight inequality at all levels. So how did you manage? And part of the answer I found in your thesis acknowledgements. Of course, you thank the team and so on, but also your Habibi, Joris, if I pronounce correctly. The person that followed you all the way to San Diego, and where during a research visit, Bart and me could also witness firsthand how Joris supports you in everything. So therefore, also on behalf of the promotion team, I would like to congratulate you, Joris and Mo, and by extension also your mother and all the rest of your family that have given you mental support from all over the globe. I would also express my congratulations to you. Dear Katherine, as I hope is clear, there are so many things that I admire you for in this achievement, and we are proud that you were part of our team, and I'm sure we'll be hearing from you because nothing can stop you. So keep going, Dr. Bassil, and congratulations.
Thank you.
Dear Katherine, Günther already mentioned most of the trajectory of your PhD, but I wanted to address you few words. So I would like to congratulate you for this nice title of doctor and this extraordinary journey that you have undertaken during your PhD and the remarkable person you have become. The journey that brought us together began after your internship in Gage Labs on the sunny beaches of California. When you came back here from you came back here not with just a tan, but also we can say that these beaches experiment have been enriched your experience and laid the foundation for your exceptional laboratory skills. From the very beginning, you exhibit a natural talent for learning and quickly became an independent force within the lab. You successfully transferred and developed several methods that you learned in the US here in Maastricht, not only the stem cells-based work, but also calcium imaging, for instance. However, it was your persistent commitment to the ethical aspect of science that truly set you apart. Your passion for infusing science with ethics has been a driving force throughout your PhD. Moreover, from reading your thesis, it is very clear that your major goal is to improve things, whether it is in science practice, mental health care, or patient well-being. During your PhD, you actively engage in teaching and mentoring, taking on the responsibility of guiding supervising students in the lab, but you quickly realized that it was not an easy way. Yet, through this experience, you grew as a mentor, learning and adapting along the way. You also learned how to manage teamwork and collaboration because of the multidisciplinary aspect of your research. And you did this very well as a great example of the nice collaboration with your parent, Rick Reinders, and Dr. Hassan Pisva on transcriptomics. I'm very proud to have been part of your journey and witness your growth firsthand. In the midst of your PhD, life presented you an extra gift, the most important gift, becoming a mother. And balancing the demand of being a mother while pursuing your career demand a lot of quality, and that you have these high qualities. So you could combine both, and you are like a superheroine that can tackle multiple tasks with ease. When we said that you never give up for everything, it was not totally true. Indeed, it was your initial plan that we speak French together, but you give up quite fast because your commitment was elsewhere in your research, in your ethics. And yeah, I think that was very demonstrating that your ability was elsewhere, in your true passion. So working with you as a supervisor has been an absolute honor and pleasure. So your wide range of skills combined with your never give up attitude made an everyday venture. I have seen your dedication, your resilience, and your determination firsthand. So your future carries is undoubtedly destined for greatness, and I have confidence that you will continue to inspire the others with your passion and dedication. And to conclude, as you said, you are not just a woman in science, but you are a real scientist. Thank you.
Dear Dr. Bassil, how does that feel?
Very good.
Okay, very good. I also would like to congratulate you on behalf of Maastricht University. And in addition, I would like to also congratulate you on behalf of Professor Gage. I met him a couple of days ago. We had a Zoom meeting about some things, and he said, bring forward to this event his congratulations. I also would like to bring the congratulations to your promotion team, who basically, the three of you, who guided you in an ethical way or in an ethical way to reach you here. And I was just sitting here and listening. Is this lady now resilient or not resilient to stress? Because I got the feeling that she's fully resilient to stress. I think so. Is that correct?
I hope so.
You hope so. No, you should be. In my congratulations, of course, I also bring forward your family, your husband with the daughter who is perfectly quiet for 1 hour. I think it's very good. And so I'd like to congratulate them as well. I would also like to thank the members of the Corona who were here today, all of you from Canada, from the United States, from Belgium, not from Belgium, from Leiden. And I think it's very much appreciated, and it gives a special flavor to this Corona that they could join and ask you all different kinds of questions. And of course also our colleague online, Professor Lesch, that he could be available for this time. So saying this, we're going to officially close the ceremony. And for closing the ceremony, I use my hammer. You haven't seen it here, but I have him right here. Yes. And with this knock, I officially close the ceremony, and we're going to continue. How are we continue? We will have a celebration. There is some drinks afterwards, which is quite logically after this event. So I would like to invite the audience already to go to the Receptor. And if you don't know what the Receptor is, just follow the mainstream and you'll find it yourself over there. There is some drinks available, some coffee, some tea, and even some